RNA is built from just 4 chemical letters, A, U, C, and G. But to build a living organism, those 4 letters need to carry the instructions for 20 completely different amino acids. If one letter coded for one amino acid, we'd only get 4. If they worked in pairs, 4 squared gives us 16. Still not enough. It took a physicist named George Gamow to solve the biological math. He argued that the letters had to be read in combinations of 3. 4 to the third power generates 64 possible combinations. That safely covers the 20 required amino acids, with plenty of room to spare. Looking at this text from the NCERT textbook, we see Gamow's 3-letter combination defined as a codon. Out of the 64 possible triplets, 61 actively code for amino acids, while the remaining 3 function purely as stop signals. This mathematical necessity forms the structural foundation of all protein synthesis on Earth. Because it governs everything from bacterial enzymes to human muscle tissue, it is a guaranteed high-yield topic for the NEET exam. This chart shows the genetic code checkerboard, the master dictionary cells use to translate mRNA into proteins. Pay close attention to the codon AUG. It carries a strict dual function. It acts as the initiator codon and explicitly codes for methionine. Here is a diagram showing the physical translation process. A tRNA adapter molecule docks its specific antihodon directly to the matching mRNA codon. If the mRNA reads AUG, followed by UUU and UUC, the tRNA physically brings in methionine, followed by 2 phenylalanine molecules. Accurately reading this dictionary is the first required step to securing your marks. The sequence of these codons dictates the entire downstream structure of the protein. When tested on this code, students chronically confuse 2 specific NCERT terms, unambiguous and degenerate. Unambiguous means strict loyalty. One codon points to one amino acid. UUU always codes for phenylalanine. Degenerate describes redundancy. One amino acid is coded by multiple codons. Unambiguous is like a strict government ID. Degenerate is like having multiple nicknames for the same person. Reversing these 2 relationships is the most common way students trigger negative marking in exams. Keep the direction of the arrows clear in your head. The genetic code is read in a continuous, contiguous fashion. There are no commas, no spaces, and no punctuation marks between the codons. Look at this NCERT text block. Consider the sequence RAMHASREDCAP. A point mutation is a substitution. Swap the E in red for an A, and it becomes RAMHASRADCAP. One word changes. This exact mechanism causes sickle cell anemia. But what happens to the reading frame if we insert a brand new letter, like a B, right here? Because the code is read continuously in blocks of 3, everything slides over. The sequence now reads RAM has bred DCA. Point mutations damage a single amino acid. Frame shift insertions or deletions destroy the entire downstream structural message of the gene. Let's test this with a rapid-fire sequence. Mentally translate this mRNA strand: 5' AUG, CCG, UAA, GCA, 3'. The ribosome starts at AUG, adds the next amino acid for CCG, and then immediately drops the polypeptide chain. The final GCA codon is completely ignored. This happens because UAA is a stop codon. Along with UAG and UGA, these 3 specific triplets do not code for any amino acids. They simply terminate the process. Here is the specific numbered list of the 6 salient features of the genetic code, straight from the source material. Take a screenshot of this list. Memorizing these 6 rules and understanding the mathematical logic of the reading frame locks in 4 guaranteed marks on exam day. Keep your definitions strict, watch your reading frames, and the translation becomes automatic.